So in part 3 of this lecture, we're going to look at lipid metabolism, so adipose tissue metabolism, and also metabolism of intramuscular triglycerides, or IMTGs. So in terms of this diagram, which we first saw in the last part of this lecture, we're mainly focusing on the left-hand side of this diagram in this part of the lecture. So in terms of where we store lipids in the body, we store lipids in the form of triglycerides or triacylglycerols in the adipose tissue, and in a healthy lean individual this can be anywhere from 5 to 10 kilograms. We also have fatty acids in the blood, which can be taken up into the muscle, and we also have a store of lipid in the muscle called IMTGs or intramuscular triglycerides, and here we have around 350 grams. And these can then undergo energy metabolism processes, again to generate ATP. So we know we need to oxidize fatty acids, but where do these fatty acids come from? And they come from lipolysis in adipose tissue and also skeletal muscle. So when we consider lipolysis, the key enzyme that we need to remember is HSL, or hormone-sensitive lipase. Now, hormone-sensitive lipase is activated by adrenaline and inhibited by insulin. So here we have our muscle containing IMTG, and here we have our adipose tissue, which also contains triglyceride. And here we have our triglyceride. So it's a glycerol backbone with these 3 fatty acid chains attached. And then through the action of HSL, we can break some of these bonds to leave free fatty acids. And these fatty acids can then be taken up, can be released into the blood to be taken up into the muscle if we're talking about the adipose tissue. But if we're talking about the breakdown of IMTGs, these fatty acids can be directed straight into the mitochondria for energy production. So to look at this lipolytic process in more detail, here we have our triglyceride, which is broken down to diglyceride, releasing a fatty acid. The diglyceride is then broken down to monoglyceride, releasing a fatty acid. And then the monoglyceride is broken down into its constituent parts, leaving glycerol and fatty acid. And HSL is important in catalyzing these first 2 reactions, so the breakdown of triglyceride to diglyceride and the breakdown from diglyceride to monoglyceride. And then MGL, or monoglyceride lipase, is responsible for breaking down monoglyceride into glycerol and a fatty acid. So to illustrate that then, here we have our glycerol backbone with 3 fatty acids attached. This is our triglyceride. Broken down to a diglyceride, we then have our glycerol backbone with just 2 fatty acids attached. Breaking that down to a monoglyceride then, we have our glycerol backbone with 1 fatty acid attached. And then through the action of monoglyceride lipase, we're then just left with our glycerol backbone. So the fatty acids that are released from adipose tissue can travel to the skeletal muscle in the blood for oxidation. However, the fatty acids that are released from the IMTGs, or the intramuscular triglycerides, can be directed straight into the mitochondria for oxidation. So this is a quick overview of the processes that are involved in fatty acid oxidation. So here we have the cytosol, or the watery part of the muscle cell. Here we have the membrane of the mitochondria, and these are the processes that occur within the mitochondria itself. So in the cytosol we have long-chain fatty acids, and through a series of reactions which are catalyzed by CPT1, for example, we have fatty acyl-CoA inside the mitochondria. This then has to undergo beta oxidation to form acetyl-CoA, and the acetyl-CoA can then enter the TCA cycle and the electron transport chain in order to generate ATP. So if we look at the processes of fatty acid oxidation in more detail, this is known as the carnitine shuttle. So in order to get the the fatty acids into the mitochondria, they have to undergo the carnitine shuttle, which involves the molecule carnitine. So in the cytosol, we have fatty acyl-CoA, and carnitine sits within the intermembrane space. So here we have the mitochondrial outer membrane and the mitochondrial inner membrane, and then this is the inside of the mitochondria itself. So CPT1 sits at the outer mitochondrial membrane, and it allows fatty acyl-CoA to bind with carnitine to form acylcarnitine and coenzyme A. But the acylcarnitine then needs to cross the mitochondrial inner membrane, and here CPT2 is important. So acylcarnitine binds with CoA. Through the action of CPT2, we are left with carnitine and fatty acyl-CoA, and fatty acyl-CoA is then involved in beta oxidation, as this is the next step of fatty acid oxidation. So this process is important in allowing this fatty acyl-CoA to cross the intermembrane space and enter the mitochondria. So to look at the two key reactions in a little more detail then. So reaction 1, we have long-chain fatty acyl-CoA plus carnitine through the action of CPT1 generates acylcarnitine and coenzyme A. And reaction 2 involves acylcarnitine plus coenzyme A. This is catalyzed by CPT2, or carnitine palmitol transferase 2, and this leaves us with fatty acyl-CoA plus carnitine. However, this is only important for the long-chain fatty acids. The short and medium-chain fatty acids can actually enter the mitochondria directly. So the next part of the lipid oxidation pathway is beta oxidation, and this involves the conversion of fatty acyl-CoA to acetyl-CoA, as acetyl-CoA is the beginning of the Krebs cycle, whereas fatty acyl-CoA would not be able to undergo the Krebs cycle. So this is a cyclical reaction which converts fatty acyl-CoA to acetyl-CoA, and the enzymes that are involved in beta oxidation are acyl-CoA dehydrogenase, beta-hydroxyacyl-CoA dehydrogenase, or beta-HAD or beta-HAD, and this is one that I'd recommend you remember, as it's one that you'll come back to time and time again in research papers as you progress through the course. And the third important enzyme is beta-ketoacyl-CoA. So the product of beta oxidation is acetyl-CoA, and this is the first step of the TCA cycle, which we covered in detail in part 2 of this lecture. So again, you can see that this TCA cycle is a cyclical series of reactions going from acetyl-CoA through citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and finally oxaloacetate. And you'll notice that this series of reactions is important in generating NADH and FADH2. So over here you can see the input into the reaction and also the output of the TCA cycle, and it's these outputs that are important in fueling the electron transport chain, which is a series of reactions which occurs through complex 1, complex 2, complex 3, complex 4, and complex 5, which is also known as ATP synthase, and it's the reactions, particularly through complex 5, that allow us to generate ATP, and generate, in terms of fat metabolism, a huge amount of ATP. So if we look at this fatty acid here, so this is the formula for palmitate. We require 23 molecules of oxygen to oxidize palmitate, and 130 ADPs and 130 phosphates. And that allows us to generate 130 molecules of ATP, as well as other byproducts like CO2, carbon dioxide, and water. But the important thing to note is that lipid metabolism allows us to generate a huge amount of ATP. It's just a slightly slower process. As you've seen, we need to undergo lipolysis, the carnitine shuttle, as well as beta oxidation before we enter those more oxidative processes like the TCA cycle and the electron transport chain. So although it's a slower process, it actually enables us to generate a huge amount of ATPs. So 130 ATPs we can generate through oxidation of palmitate.